This is what we're going to talk about next. It's direct transmission, indirect transmission. So we talked about direct transmission. So for indirect, understand that what is nosocomial infection is and how it works. Okay. Remember that's hospital-acquired infection. Indirect transmission. What can we have as an example? Through the fomites, right? Through the fomites, through the, say, aerosolized, like urine or aerosolized droppings. By the way, every time you go to the restroom and you do your things and then you flush, you aerosolize several millions microbes and you inhale them, by the way, just in case. Okay? So if you have a restroom that is close to the kitchen, keep the door closed. Okay? Just a piece of advice here. Cox postulates. Understand why it's so hard to fulfill them and understand how it works. Okay? So that you gotta have isolate. Sometimes you can't. You gotta culture. Sometimes you can't. You gotta find a model. Sometimes you can't. So it all kind of puts certain restrictions on the Cox postulate. Epidemiology. Understand the difference between these four. Okay, we discussed it quite details. Okay, if you have any questions, ask now. But know the difference: sporadic, endemic. Enzootic, the disease that occurs in animals in a certain region. So it's endemic in people, enzootic in animals. Like rabies, enzootic in the United States. There are rabid animals out there. Reportable disease, just know that it's the one that you need to report to CDC. You saw that gigantic list. You don't have to memorize it. Although, of course, I'm tempted every time to say that you have to. No, you don't have to. Don't worry. Difference between incidence and prevalence. Incidence is the number of new cases. Prevalence is number of sick people in the population. Difference between morbidity and mortality. Morbidity is how bad the disease is, and it depends on the infectious dose. Mortality is how many people die, and high mortality is usually associated with a lower LD50. Okay, so less microbes are necessary to cause mortality. Okay, let's talk about innate system and immunity. So here we have two lines of defense. Internal defenses. So immune cells, what I mean, those, yeah, 3 granulocytes, 2 agranulocytes, right? So eosinophils, basophils, neutrophils versus granulocytes, monocytes, and natural killer cells, agranulocytes. For each of the cells, know mechanism of defense and what they act against. Okay? Please review that. You can use the study guide, kind of make a little table next to it and just write briefly. Something like eosinophils, parasites, fungi, allergy, mechanisms, cellular toxicity, inflammation. That's it. I have a table. It's I think it's the second slide of the lecture that summarizes all of that. Inflammation, 5 signs. Again, we talked about that. Steps of the response. Now look. So release. So leukocytosis. Okay. Leukocytosis. Understand the sequence. So you have leukocytosis first. Migration to the site of inflammation second. Understand the steps. Like white blood cells are produced, right? They go to the site of inflammation. They migrate through the wall in extravasation, extravasate through the wall, and then they follow positive chemotaxis to the site of infection. Antimicrobial proteins, 2 types of interference. Can anyone name them? One and 2. There are 2 types of interference, type 1 and type 2. Okay? When I say production and signaling, it may sound scary, but what we discussed actually is that interference is produced in response to the infection, right? So you got to get infected to produce interference. And then they signal on the same cell, autocrine, adjacent cells, paracrine. or distant cells, endocrine. Mechanism of defense. What do they do? Correct. They warn other cells to start producing antimicrobial proteins, right? So in essence, they essentially stimulate certain genes to start getting expressed and produce proteins. The targets for interferon, we didn't really discuss. Viruses and bacteria, that's all you gotta know. Viruses, bacteria mostly. Complement, location in the body, where it is in the body. It's in the blood, right? Now, the activation cascade. Activation cascade. Principle here is that you have 3 different pathways. For each of the pathways, stimuli, and that they all come to the common intermediate, which is C3 protein, okay, and the effects. Inflammation, okay, phagocytosis, sorry, it's Russian. It's Russian letter F. Phagocytosis, okay, and membrane attack complex, okay. Adaptive immunity. Understand that it's specific. It is linked to the innate system via enhancement of inflammation, and that it activates complement. Via what? Antibodies, right? Now, again, 3 pathways. Know them. I'm going to ask you, what is the stimulus for the pathway? Classical, alternative, lectin pathway. What initiates those pathways? Properties of the immune system. Specificity, what does that mean? It is mounted to a specific antigen, right? And it has immunological memory. Okay, it has a memory. Antigens. Understand the difference between the complete antigens and incomplete. Complete antigen is what? Big and complex, right? Incomplete, we call it haptene. How can we produce antibodies to haptene? Huh? We can stick it to big molecule, like big carrier, right? So they kind of look big. Understand the concept of antigenic determinants. Each antigen, like a protein, how many antigenic determinants can it have? One word, 4 letters, starts with M. Many. Okay, makes sense so far? Mhc proteins. This little topic here is probably the hardest part. Understand the concept of major histocompatibility complex, where they are type 1, type 2, okay? Type 1, type 2. Type 1, so that's MHC restriction. CD8 recognized type 1, CD4 recognized type 2. But type 1 is on all nucleated cells. So it's like IM cell. If it, so, yeah, MHC class 1, I'm fine. But if it presents the foreign antigen, then, dude, I'm infected, kill me. Okay?
So, like, would it be a foreign antigen, or would it be like, I understand the cancer thing.
Oh, of course, cancer cells are abnormal. They present antigen that is abnormal, foreign, okay? That doesn't look normal, right? Type 2 can be found only on antigen-presenting cells like B cells, macrophages, and dendritic cells, for example, and it's specifically recognized by CD4, okay? Now, cells of adaptive immune system, effector cells. Remember that B cells themselves don't produce antibodies, okay? Plasma cells produce antibodies, okay? Plasma or helper or killer or regulatory. Memory cells, just understand the concept that all subsets produce memory cells. Concept of VDJ recombination. The variability of receptors that recognize antigen is provided by VDJ recombination. Remember we talked about it, like different genetic fragments? They assemble like Lego, yes.
What is that you want us to know about?
That we have never numbers. Never numbers. You don't have to memorize this meaningless numbers. There are V genes, D genes, and J genes. You combine those fragments, and those fragments form variable region of the antibody molecule or B cell receptor molecule or T cell receptor molecule, whatever it is. And then because there's such a huge combination, the number of combinations, they can recognize any antigen. Okay? Questions on that? For T cells, maturation in thymus versus maturation in B cells. Maturation in bone marrow. Thank you very much. And positive negative selection. So positive selection. T cells are selected for ability to recognize MHC. Right?
So it's like
MHC. Huh?
like immunocompetency?
Yes. MHC recognition. Negative selection is no self-recognition. Does that make sense? So they're not autoimmune. Does that make sense?
So what happens to ones that are negatively selected?
If T cell recognizes the own antigen, it goes into apoptosis. Does that make sense? Self-antigen goes into the apoptosis. For secondary lymphoid organs, all you have to know is that B and T cells go into the lymphatics. Period. Antigen-presenting cells. Macrophages, dendritic cells. B cells can be considered APCs, but I'm not going to trip you over in that. Let's do this. Let's just pause this, and while it exports, we're going to